Hello, everyone, and welcome to our next lecture topic. Today, we're going to be talking about DNA structure and replication. So up until this point, for the past couple of weeks, we have actually talked a lot about DNA, even going back to when we were discussing cell structure. We talked about how DNA is housed inside of the nucleus. And during our discussions of mitosis and meiosis, we talked about how the cell has to replicate DNA before it can go through those processes, because again, we're making different daughter cells that have either identical copies of DNA or have genetically different copies of DNA. So we've discussed DNA a lot, and now it's time for us to actually understand a little bit more carefully what the structure of DNA is and actually how we make those copies and how we replicate it. So the beginning part of the lecture is a really brief history of how we discovered the structure of DNA. I'm going to go through that very quickly. What I really want to focus on in today's lecture is the actual structure of DNA itself and how DNA replicates. So since the late 1870s, there have been a whole host of different scientists whose work has really led to our understanding of what DNA looks like and how DNA replicates. So we had some early pioneers in this field, Miescher, for example, who was able to isolate this acidic substance from white blood cell nuclei. He called it nuclein. We later referred to these as, nucleic acid, which again, that's deoxyribonucleic acid. That's where we get the name of DNA. There were scientists like Frederick Griffiths and Hershey and Chase who did different types of experiments with either bacteria or viruses, where they were trying to determine whether it was the protein part of bacteria and viruses, or whether it was the DNA part of the bacteria and viruses that was responsible for passing along that genetic material. So Griffiths did an experiment with mice and bacteria. Hershey and Chase did an experiment with viruses and bacteria. But the conclusion for both of those experiments, and you can read a lot more about them in the text, is that it is not the protein that surrounds the bacteria or the protein that surrounds the virus, but it's actually the material inside, the DNA inside, that is actually responsible for passing along that genetic information. So once we understood that DNA was the hereditary molecule, then we sat to figure out, okay, so what does DNA look like? What's its structure? And Phoebus Levine was a Russian-American biochemist who was the first one to identify nucleotides. And he figured out that a nucleotide consists of sugar, a phosphate, and a base. So he knows that DNA is the molecule. DNA is made up of subunits, and those subunits are made up of these three parts: sugar, phosphate, and base. Continuing on with that work, we had Chargaff, who was an Austrian-American biochemist, and he was actually able to determine that those nitrogenous bases are either adenine, guanine, thymine, or cytosine. So he was actually able to come up with the different types of bases that we see. Then we have most notably the work of Rosalind Franklin and Maurice Wilkins. These were English scientists, and they were able to basically figure out that there were kind of two forms of DNA. They had found this A form, which was very dry and very crystalline, and then they found this B form, which was very wet and cellular. And as it turns out, the B form was the form that was one that they wanted to capture an image of. Rosalind Franklin in particular was working with a technique called X-ray diffraction. She had used it successfully in other things that she had studied, and she began to sit down to try to figure out and take a picture of DNA using this X-ray diffraction technique. This was a very long and very complicated process she went through. It took her about 100 hours to obtain like the perfect photo of the B form of DNA. And unfortunately, her work didn't go much farther beyond there because she died at a very young age of cancer because of all of her work with X-rays. We didn't understand at the time that X-ray radiation is very damaging to our cells, and it's a known mutagen which can cause cancer. So she passed away at a very early age of cancer and wasn't really able to continue her work much beyond the pictures that she took. However, two other gentlemen, James Watson and Francis Crick, saw her pictures and they said, hey, I know exactly what DNA looked like. And so they took her work and Maurice Wilkins' work, and then they were able to create a 3D model for everyone to show them exactly what DNA looks like. So they themselves didn't perform any of these experiments, but they built off the knowledge of other folks. And so unfortunately, for the longest time, these two gentlemen were given basically the discovery of the structure of DNA. Everyone said, oh, it's Watson and Crick and Watson and Crick. Later on, thankfully, we began to remember those other folks who were instrumental in the discovery, including Rosalind Franklin and Maurice Wilkins. So, nice little table here that sort of summarizes all of that information that I just told you and the timeline of discovery, if you want to think of it that way. All right, let's get into the more important thing here. Let's talk about the structure itself. So DNA is made up of building blocks. Each building block is referred to as a nucleotide. A nucleotide has three pieces: a deoxyribose sugar, a phosphate group, and a nitrogenous base, of which there are four: adenine and guanine and cytosine and thymine, and we'll worry about purines and pyrimidines here in just a moment. All right, so here's what the nucleotide structure looks like. So for every nucleotide, we once again have a sugar, a phosphate group, and one of four different types of nitrogenous bases. We can classify these nitrogenous bases based on how many rings that they have, how many, if they're single ring or double ringed. Our double ringed bases are referred to as purines. This is adenine and guanine. And our single ring bases are referred to as pyrimidines. So this is cytosine and thymine. So here we have the beginnings of a molecule of DNA. We're going to start with one strand. So DNA is actually a double-stranded molecule, but let's just focus on a single strand of DNA here for a moment. And what you can see are each individual nucleotide here. So here are the nucleotides sort of stacked on top of each other. And we have these sugars that are connected to each other with a phosphate. So it's a sugar, phosphate, sugar, phosphate. It creates this continuous backbone, as we call it. And then sort of sticking out here into the middle, we have our nitrogenous bases, right? Adenine, guanine, cytosine, and thymine, which we represent with the letters A, C, G, and T. So now, again, DNA is not single-stranded, it's actually double-stranded. So we're going to put two of these strands together, but what you should notice is there's something a little bit different about this second strand. It's not exactly parallel. You'll notice that all of the letters on this side are right side up, and all of the letters on this side are upside down. That's intentional because we have an opposing orientation. It's a head-to-toe orientation. We call this anti-parallelism. So DNA is double-stranded, but it's an anti-parallel molecule. So we have one strand that's sort of flipped up on its head, and it runs in the reverse order. So this image of the two hands is just kind of describing that anti-parallelism. So I don't want you to worry too much about sort of how we determine the 5' to 3' and the 3' to 5' nature of how we're orienting these strands of DNA. The information is in here in case you want to know exactly how we are referring to DNA, but I'm not going to expect you to know this on an exam. It's enough for you to know that one strand runs in what we're going to call the regular direction, and the other strand runs in the opposite direction. So you can think about it kind of like reading a book. So when we read a book from left to right. Okay, so you can think of this left strand as being the correct way to read DNA. So starting at the top and moving down to the bottom. That's the correct way we read it. The other strand of DNA is backwards. So if we were to read it from top to bottom, it would make no sense to us. It would be backwards. So we actually have to read it from bottom to top, as it turns out. Now, that's going to be more important here in just a minute. So our base pairs, again, we said the complementary base pairs are A's and T's, C's and G's, right? Adenine goes with thymine, cytosine goes with guanine. How these pairs are held together are through the formation of hydrogen bonds. So we have hydrogen bonds that are forming in between our pairs of nitrogenous bases, and that's what actually holds the two strands of DNA together. So again, A's always pair with T's, C's always pair with G's. Just because of the structure of how these look, an AT pair will always have 2 hydrogen bonds, and a CG pair always has 3 hydrogen bonds. Again, you don't really particularly need to know that, but in case you're interested. When we were talking about DNA inside of our cells and we were talking about how DNA goes through the process of replication, I talked about how when it's loose in its what I called spaghetti form, it was nice and loose and it was really easy to access all of the genetic material, right? Those genes. We can get to those genes because spaghetti is loose. But when we moved into talking and discussing the process of cell replication, we said that that loose DNA had to condense and those chromosomes had to form. So this is what that process actually looks like. In order to condense, what DNA does is it coils around these proteins called histones, and it forms kind of like a bead on a string structure. The bead part is called the nucleosome, and then the nucleosomes in turn will wind up tighter and tighter and tighter, forming what's called chromatin. And then that chromatin then can be condensed in what we see as chromosomes. So again, here's our naked DNA. The DNA wraps around these histones. When the DNA is wrapped around a histone, we call it a nucleosome. When we spin these things together and wind them up, we refer to this as the chromatin. And we can see these nice condensed chromosomes when we're going through the process of DNA replication. Excuse me, not DNA replication, when we're going through the process of cell replication, right? When we're going through mitosis and meiosis. We need that DNA to be uncondensed, actually, when we go through DNA replication. So just as a reminder, gene is a segment of DNA. It directs the formation of RNA to produce proteins. That's next lecture's topic. And then the protein is responsible for the phenotype or the trait that we see. And all of this information, how to make a protein, is conveyed by the sequence of nucleotides in DNA. And again, we're going to talk about that in our next topic. But this sequence of nucleotides, the specific order of A's, C's, T's, and G's, is very important. So let's talk a little bit about DNA replication just here for a minute. Once again, it occurs in the S phase of the cell cycle prior to cell division, either mitosis or meiosis. In humans, we can replicate about 50 bases per second. And what's happening is in a human chromosome, we are replicating simultaneously at 100 of points along its length. So it's not like we start at one end of the chromosome and we work our way all the way down to the other end of the chromosome, which can be, 1,000, tens of thousands of base pairs long. No, what we're doing instead is we're doing it at multiple points. And any place that we do this is referred to as a replication fork. So here's the quick and easy version of DNA replication. We have a DNA parent molecule. We have to unwind it and separate the two strands, and we do this at several different points along a chromosome. Each parental strand is going to serve as a template for replication. Then a molecule called DNA polymerase is going to bind the complementary base pairs to all of our open nucleotides here. So everywhere where we see an open nucleotide,
A DNA polymerase is going to come and it's going to grab a free nucleotide and actually just pop it right on there. Excuse me, a free base pair, not a free nucleotide, a free base, and it's going to pop it on there. Okay, so then we're going to wind the whole thing back up and we're going to move on. Okay, here's some different enzymes that are important in DNA replication. Again, I'm not going to go through these extensively, but we have enzymes that unwind the DNA, enzymes that bind to it and stabilize it, ones that add short sequences like leader sequences. We have DNA polymerase is the one that we're going to discuss the most. This is the one that binds those new nucleotides, and then ligase, which joins them all together. So here we've got our replication fork. Really the only thing you need to know is in one direction we can continuously do replicate DNA. So the 5' to 3', we can have the enzymes here working just boom, 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 adding those nucleotides 1, 2, 3, 4, 5 after another. Great, no problem. On the other side, we can't do that. Remember, it's backwards. So we have to work in these little tiny fragments. We do it one piece at a time, and then we jump ahead and we work backwards again. And then we jump ahead and we work backwards again. And we do that over and over and over again until we have all of those letters bound with complementary base pairs. Okay, so again, DNA polymerase is going through, whoop, replicating in one continuous direction. The other direction, it will replicate in fragments, which are called Okazaki fragments. So there's the Okazaki fragments, and then you have to put them together. Okay, so again, then we bind everything up, ligase seals it all up, and voila, we are left with 2 identical copies of our DNA molecule. Okay, so that's the quick version of it. Again, I highly recommend going into your text. There's actually at the end of that section, there are some videos that'll show you a little bit more about DNA replication. Highly recommend those as a reference. For today's lecture, you really should know what a nucleotide is, the difference between purines and pyrimidines. You should know what a double helix is, understand what a nucleotide is, what the nitrogenous bases are, and how they pair together, and be able to explain why DNA is considered semi-conservative. Oh, I guess there wasn't a particular slide about this. I didn't realize that. Okay, here at the end, I'll tell you about semi-conservative replication. It's called semi-conservative replication, excuse me, because each strand of replicated DNA contains one of the original parent strands that we started with that acted as a template, and it also contains one newly assembled strand. So it's semi-conservative because we're using one strand as a template and the other strand we're building from new. So there you go. I'm not sure why that wasn't in my notes. I might have to go back and refine that. Thanks, everybody.